Hi, Amirreza here. I'll be presenting our work titled Gene Network Connectivity Conveys Robustness in Gene Expression across individuals, cell types, and species. This is joint work with Olga Sigalova, who is currently a postdoc at EMBL, and my supervisor, Judith Zog, who is a group leader at EMBL. Okay, living systems have a remarkable capacity to be robust against different sources of variations, such as genetics, environmental, and stochastic. On the other hand, modern statistical and machine learning methods often tend to be non-robust. More precisely, first, these methods are usually very sensitive to the corrupted data. Second, adding a small adversarial perturbation to their inputs leads to misclassification with high confidence. As it can be observed, adding an imperceptible adversarial perturbation to the image of a pig can easily fool modern deep nets to classify it as an airliner. Though, indeed, this robustness property of living systems is not something trivial. In this work, we aim to better understand this property from the viewpoint of gene expression program. More specifically, we are interested in understanding the buffering mechanisms of gene expression. To be more precise, what factors are controlling the shape of a gene's distribution? Since nearly 2 decades ago, a growing body of work has been extensively studying this topic under dissimilar names, including gene expression variation, variability, and noise. In particular, our previous study reveals genomic features that make a gene highly responsive to any type of variation. However, previous publications, while shedding great light on the study of this topic, have several limitations. Majority of prior work only focused on statistics such as mean, median, standard deviation, variance, and CV of gene expression distribution. To the best of our knowledge, methods with the provable guarantee for making the median and variation statistically independent are missing in the distribution-free sense. A comprehensive and comparative approach to study the variation across different tissues and cells is missing. In previous studies, the connectivity of genes through the networks is usually not taken into account. Prior work has not much focused on evolutionary aspects of variation. And finally, most studies have overlooked the proteomics variation. In this presentation, we will only focus on some of these aspects. In this work, we mainly consider data from GTEC, GWADIS, and HIPSCI projects to cover different tissues and 2 cell lines, namely B cells and pluripotent stem cells. Considering a mild assumption on a distribution, its moments can uniquely characterize it. Moreover, skewness, which is related to the third moment, and kurtosis, which is related to the fourth moment, can capture some properties of the distribution as illustrated in the figure. In this work, we aim to study 4 statistics, namely median, CV, skewness, and kurtosis of gene expression data. First, we can look at the 6 possible pairwise relationships between these statistics. Our results indicate a consistent pattern over tissues and cell lines. More specifically, median and CV are correlated with each other, which is a well-known observation. Also, skewness is not much correlated with the median, which agrees with the results of Church et al. Moreover, kurtosis also is not much correlated with the median. In addition, both skewness and kurtosis are correlated with the CV, and they are highly correlated with each other. Perhaps surprisingly, we can predict kurtosis very accurately by considering the median, CV, and skewness as our features, suggesting that the information of it is captured by median, CV, and skewness. Thus, we will only focus on these 3 statistics. Bars are showing the median, minimum, and maximum R2 over different tissues and cell lines. To start discussing our results, I briefly mentioned the main setup in the scope of this talk. We have 48 tissues from GTEC and 2 cell lines. We consider residual CV and residual skewness as the main variables that we are interested in. In fact, to have any meaningful conclusion about the properties of CV and skewness, we need to make sure that they are statistically independent with the median and CV respectively. We then take the mean of these residuals across tissues and cell lines to have an overall view. We also rely on the aggregated co-expression network across different datasets of a prior publication. Now, we are ready to discuss some of our network results. Generally speaking, statistics of a node are related to average statistics of its neighbors. Moreover, the effect is stronger for the residual CV. This is illustrated in the figure. In graph theory, indicators of centrality assign numbers to nodes according to their position in a graph. For example, we have eigenvector centrality. First, graph centralities of a co-expression network are correlated with each other. More importantly, graph centralities are correlated with residual CV and neither median nor residual skewness. As it can be observed, we have some Spearman correlation coefficients more than 0.5 between centralities and variation. Graph clustering or community detection methods reveal more separated groups for the residual CV than the median, and for the median than the residual skewness. Roughly speaking, these results can be achieved by using different graph clustering algorithms, although number of clusters might be different. Our results indicate that gene co-expression networks indeed play an essential role in controlling the expression variation. Next, I discuss some of our prediction results. To the best of our knowledge, we built the most inclusive gene-specific regulatory features characterizing properties of gene promoter, enhancer, 3' UTR, and etc. In this talk, we focus on predicting mean residual CV. It is possible to predict the variation based on only the sequence of the promoters. For this purpose, we used convolutional neural networks. As an ongoing idea, we are working on considering very long sequences using the recently published methods. Neural tangent kernel (NTK) demonstrated the best performance when we want to predict the variation based on our features. This method beats old champions such as random forest and gradient boosting. In fact, one of our contributions is popularizing this method in the genomic community. Incorporating the network into the prediction pipeline indeed improves the performance. Moreover, the improvement can be achieved by using a graph convolutional neural network model. Also, adding some additional features related to the graph structure can further a bit improve these results. Best performance in each track is demonstrated in the figure. As we can observe, we achieved more than 0.7 R² using our graph neural network model. As a final remark, I note that if we randomly permute our feature vectors, we cannot get any meaningful R² from these methods. To study the evolutionary aspects, we consider RNA-seq data from BGEE dataset. Expression variation is significantly correlated across different tissues of same species. We observed it for guinea pig as well as human. We analyzed expression variation of ortholog genes in 5 species. Moreover, we observed expression variation is conserved for closer species in matching tissues, namely tissues related to the brain. We also observed similar results as in the network part for 8 additional species, suggesting that our findings are robust across species. Here we can observe graph centrality results for chimpanzee. Next, I discuss proteomics variation. We consider proteomics data from GTEC and ISMB projects. In proteomics data, we also have a clear dependency between proteomics median and proteomics CV. Thus, we need a correction similar to the RNA-seq data, and as a result, we will have proteomics residual CV. First, we have not observed a strong correlation between proteomics residual CVs from different tissues. Second, and perhaps more importantly, proteomics residual CV is not much correlated with the RNA residual CV in different tissues and ISMB data. This is illustrated for one of the GTEC tissues. Moreover, it is actually well known that the RNA median is not highly correlated with the proteomics median. However, for the residual CV, the correlation is even lower. Okay, to summarize, we define, analyze, predict, and interpret statistics related to different moments using a variety of tools from statistics, machine learning, and graph theory, which may be of independent interest. Also, our results indicate that gene networks play an essential role in controlling the expression variation. Additionally, we observe expression variation is generally conserved for closer species in matching tissues. Finally, we have not observed a significant correlation between RNA and proteomics variation. In the end, again, I would like to thank Olga and Judith. Without them, this work would not have been possible. Additionally, I thank Mattia and Eileen, collaborators of our previous paper.